Let us now discuss few applications of SciLAC briefly. The SciLAC method is very promising for any cell line. So, this method can be applied for any cell lines, whether it is HeLa cell, C127, HEK293 or different type of cell lines people have shown. However, the media formulation and the growth optimization is required individually for each cell line. SciLAC applications have been demonstrated in different applications such as cell signaling, studying the induced protein complexes, studying temporal dynamics, identification of kinase substrates, studying differential membrane proteomics. So, there are various applications. We will have a look on some applications now. So, Ong et al. In 2002 published a paper in Molecular Cell Proteomics, which was the first SciLAC application demonstrated, where they used the relative quantitation of changes in protein expression during the time course of myoblast differentiation in mouse cells. Researchers have reported various unique metabolic labeling strategies. For example, by using tyrosine, the identification of tyrosine kinase substrates using 13C tyrosine. Labeling is also performed by using methionine, the in vivo methylation sites by heavy methyl SciLAC. There are numerous studies based on the global protein expression profiling using SciLAC method. I am just highlighting some of the very earlier studies which set up the path for performing these protein expression profiling. So, a study by Everly et al. In 2004 analyzed the expression level of more than 440 proteins in the microsomal fractions of prostate cancer cells with varying metastatic potential. Another study by Gu et al. Investigated the early stage of apoptosis by inducing the p53 upregulated modulator of apoptosis. SciLAC has also been used for functional assays to study the protein-protein interactions. A study by Blagoy et al. Used the differential labeling of proteins in EGF stimulated versus unstimulated cells. A study by De Hoog et al. Did quantification of proteins interacting in an attachment dependent manner with focal adhesion proteins. These are just few examples of studying the functional assays and performing protein interactions using SciLAC. The identification of proteins which are enriched in specific cellular structures. A study by Foster et al. Used the first functional proteome analysis of rafts, and they showed the specific detection of proteins depleted from the rafts by cholesterol disrupting drugs. SciLAC has been widely used for multiplex analysis to compare the cellular states. Anderson et al. Showed the quantitative analysis of proteome of human nucleoli. Blagoy et al. Performed a temporal analysis of the phosphotyrosine dependent signaling networks to compare the proteome of 3 cell populations. Kratchmar et al. Analyzed the divergent growth factors in mesenchymal stem cell differentiation. These are just few examples of multiplex analysis. Now, if you look into literature, there are many studies which have used the SciLAC method for comparison of cellular states. SciLAC method has also been used to study the protein turnover. Study by Pratt et al. Used the rate of breakdown of individual proteins by analysis of the mass shift in tryptic peptide fragments. The analysis of the abundant proteins in glucose limited yeast cells which were grown in the aerobic chemostat cultures at steady state was performed by using SILAC method. SILAC has been used for identification and quantitation of protein post-translational modifications. Study by Ibarola et al. Identified and quantitated phosphorylation sites. Another study by Balef et al. Also identified and quantitated the phosphorylation sites. So, there are many studies which have used SILAC method for studying post-translational modifications. So, interestingly, now SILAC method has been used in different organisms, in bacteria, in yeast. These were the more commonly used SILAC methods due to the growth in the cell culture, but there are some studies on the Arabidopsis in the plants as well as in the mice, which has shown that SILAC can be applied to the wide variety of organisms.
In an experiment conducted by Kruger and colleagues, a set of mice were fed with diet containing natural lysine. Another set of mice were fed with diet containing C13 labeled lysine. The researchers tracked the incorporation of lysine 6 into the mouse proteome over 4 weeks by providing the C13 containing lysine diet. Their developmental growth and behavior were observed in addition to sampling various blood proteins. The labeled mice were found to develop normally. Average lysine 6 incorporation over 4 weeks in human serum albumin and hemoglobin is depicted in the graph. D. Godoy and colleagues determined fold change of peptide pairs between haploid and diploid yeast cells using SILAC. Labeled lysine residues were used to grow the diploid yeast cells, while haploid cells were grown in normal lysine medium. The cultures were mixed, proteins extracted and analyzed by LC-MS. Protein ratios between haploid and diploid cell were determined with high accuracy. Comparison revealed 97.3% of proteome to change less than 50% in abundance between haploid and diploid cells.
Let us briefly look at the eye track applications, and I will show you this in this animation. In this animation, we will look at one application of eye track method. A study performed by Boyle et al. In 2010 used the eye track method for identification of candidate biomarkers in ovarian cancer serum. So, serum samples of control and cancer patients were first of all depleted by using a multiple affinity removal system to carry out immunodepletion of the serum samples from normal controls and ovarian cancer samples. This step helped in removing the high abundance proteins, leaving behind only the medium and low abundance protein for eye track analysis. The immunodepleted serum samples were then labeled with eye track reagents and further analyzed in MSMS. The authors detected a total of 220 unique proteins, of which 14 were found to be elevated in ovarian cancer serum samples as compared to the healthy controls, and 4 novel candidate biomarkers were first time reported. These results were further validated by the Western immunoblotting. This just gives you an overview of how eye track reagents can be used for various type of applications, including in biomarker discovery.
IIT has found many applications for proteomics studies. In one such clinical application, Kang and co-workers obtained plasma proteomes from 6 breast cancer patients and 6 healthy controls. These plasma samples were first treated on an immune affinity column in order to deplete them of their high abundance proteins. The immunodepleted serum samples were then used for further analysis by IIT. These samples were first treated on an immune affinity column in order to deplete them of their high abundance proteins. The immunodepleted serum samples were then used for further analysis by IIT. 155 proteins were identified, of which 33 showed 1.5-fold abundance change in plasma of breast cancer patients as compared with the healthy controls.
In 2010, Human Proteome Organization (HUPO) has launched a Global Human Proteome Project (HPP). This project is designed to map the entire human proteins encoded by the genome. Let us now discuss some of the targeted focused initiatives. The Human Liver Proteome Project. This is the first initiative of Human Proteome Project for human organ, tissues with an intention of generation of comprehensive protein atlas of the liver and international liver tissue network, collection and distribution of normal liver samples and validation of new discoveries. Plasma Proteome Project. Analysis of the protein constituents of human plasma and serum. Human Brain Proteome Project (BPP) focuses on the revelation of the brain related proteomic alterations, focusing on understanding neurodegenerative diseases, aging and identification of prognostic and diagnostic biomarkers. Human Kidney and Urine Proteome Project aims to understand kidney functions, mechanism of chronic kidney diseases at a protein level and discover biomarkers and target molecules for new therapeutics of kidney diseases.